Good morning, everyone, and thank you to ARM for having us speak today. We look forward to giving you an update on our progress. I'll be making some forward-looking statements today. If I can get the button to work. I am. Yeah, it's not advancing. There we go. Okay, good. Okay, I'll be making some forward-looking statements today. And start with some company highlights, a little bit about our platform. So if you're unfamiliar with Fibrocell, we're an autologous cell and gene therapy company. We focus on diseases of the skin and connective tissue, and we have an autologous fibroblast platform around those products. And we have three that are in development. Our most advanced, and the one I'll be focusing on today, is FCX007, which is our therapy being developed for recessive dystrophic epidermolysis bullosa, or RDEB. It's a rare blistering disease in children, and I'll elaborate on the disease and our approach for this. We have two other programs also in development. One is called FCX013 for linear scleroderma. This is a sclerosis disease, an autoimmune reaction of the skin that can cause these lesions that transverse over joints or over the head or over other parts of the body. They can not only aesthetically, they aren't pleasing, but they can also cause issues with formation of joints and limbs because of the fact that the skin is so sclerotic. They can't develop properly around them, and also mobility is an issue for a lot of these patients. So we have a gene therapy approach for this. We completed a proof of concept study back in January 2006 that showed some positive results. So we've completed a pre-IND meeting with the FDA, and we're planning a IND in 2017, the fourth quarter of 2017. And we just recently got orphan status for this as well. And we also have another program in arthritis and other related joint conditions. In this case, we'd be treating these joints locally instead of systemically, directing proteins of interest to those particular locations. But for today, as I mentioned, I'll be focusing on 007, and all these programs we work on in collaboration with the Exton Corporation. So let me talk a little bit about our platform, a little more detail across all the indications. So fibroblasts are the most common cell in skin and most connective tissues, which makes them an ideal vehicle for us to deliver genes of interest to those areas locally. And we do this mainly because the skin is a less vascularized area, so we're not talking about a full systemic treatment here. So there are some safety advantages we feel compared to other therapies when we're developing our toxicology profiles. And also, because they're autologous, there's obviously inherent reduced rejection immunogenicity concerns about the way we approach this. The vector of choice we are using is lentivirus. It's a third-gen self-inactivating lentivirus, which is fairly common for some of the other work being done in this area for lenti, using that third generation. The reason we chose this is the genes that we're targeting are typically large ECM proteins or something along the lines. I'll be talking about one in particular called type 7 collagen. It's a 9 kb insert, so things like AV and other vectors are usually eliminated immediately right on their packaging capacity. So it helps us with packaging size, and it also, lentivirus integrates dividing and non-dividing cells. So for a chronic disorder like RDEB or some of the other skin disorders we're targeting, it's ideal for longer-term expression, and that's what we want to show in our clinical trials. These cells are manufactured and modified ex vivo. So we think that the ex vivo approach gives us the opportunity to do some release testing. We're not directly injecting virus, so we don't see the host immune response, because in our case, we would like to repeat dose these patients because their blisters can emerge in other parts of their bodies and other localities. So we think there's some advantages of our approach from a manufacturing standpoint. And obviously, we're using our regulatory experience with our non-modified products and our manufacturing experience to leverage onto these programs as well. So just a little bit about the process of manufacturing itself. We take biopsies are collected from the patient, small biopsy punches. They're sent to our manufacturing facility. For the 007 program, we're working with PCT out in Mountain View, California. They'll expand to low passage in vitro, and then we perform the transduction activities at low passage. This allows the cells to divide once they've been introduced with lentivirus, and introduces our construct, and then they divide and obviously carry them along to progeny cells as they move into larger populations, and then we'll harvest those. They're then cryopreserved, and then when the patient is ready, they can be thawed, washed, and shipped for intradermal injection the next day. So off the platform and onto the disease a bit. If you're not familiar with RDEB, it's a monogenic disorder of the skin where there is a mutation in what's known as the COL7A1 gene. This encodes a protein known as collagen 7, type 7 collagen. And the reason that's important is it makes up the what are called the anchoring fibrils. And I think the laser pointer is working. But if you look back at the image on the left, the normal skin, you see these sort of arcing structures that are coming from the dermal epidermal junction down into the dermal layer. These are anchoring fibrils, and they're primarily formed from collagen 7. So in the event you're not properly forming those fibers in this area, you get a situation where they're not connected properly between the two layers of skin, and you essentially have two layers of skin that are floating on one another. And just a small amount of lateral friction then causes a blister void to occur. It then fills with liquid, and unfortunately, if pressure occurs downward on that blister, it spreads laterally. So you can imagine that pretty quickly you start accumulating blisters on the body. And right now, the standard of care for this, obviously, it's devastating, it's painful. Standard of care right now is really just palliative. It's blister care, cutting blisters early so they don't spread, treating them with bandages, antiseptic treatment, and obviously there's a lot of surgeries involved, namely finger separation surgery is common, and also esophageal dilatation affects the oral mucosa as well. There is a high mortality rate to this disease, even though it is a blistering disorder. It comes from squamous cell carcinoma primarily, and then sepsis is usually the second leading issue, and typically by their mid-20s, that's typically when they succumb. So it is a very, very terrible disease, an expensive disease for families, especially if there's coverage up to $10,000 a month just in bandage and wound care alone. In the epidemiology, obviously, you can see that's an ultra-rare disorder of about 1,100 to 2,500 patients U.S. So you can see in this photo this young lady here, that's a typical patient. The bandaging is it's preventative, trying to prevent sort of blistering issues going on with their limbs, where they tend to see most of the activity of wounds. So just in general, the idea here is we have a monogenic disorder with a structural protein, and our goal is to take an autologous human dermal fibroblast and transduce that with the COL7A1 functional gene synthetically made, and then re-administer that to the wounded areas of patients. So the next two slides, I'll talk a little bit about the data that we've collected, sort of the early proof of concept and pharmacology data that supported our IND filing to the FDA. First of all, in a lot of the academic literature, the structure of collagen 7 is obviously very well known. It's a triple helix structure. They form monomers on the monomer chains over here on the left at about 290 kilodaltons. They'll then form a triple helix, as collagens do, and then they anti-parallel, and then they bunch together to form these anchoring fibrils. So, what we went out and did with Entrexon is develop a number of characterization assays to show that not only is it structurally or reproducing collagen 7 and we can detect it, but is it the proper size and functionality? And one of the important aspects is forming this triple helical trimer. So when we looked at a lot of the academic literature, we noted that on immunoblots from Western blotting and immunoprecipitation, you see heavy monomer chains in a lot of their work. So we worked hard to optimize to make this primarily trimeric. So what you're looking at are bands up to 900 kilodalton weight ladder, along with purified collagen 7, so a standard purified, and then our products, and then here's an RDEB patient culture, the supernate, and as we can see, in our gene-modified lanes, we're seeing this trimeric primarily collagen. This was very promising to us because that suggests it's in the proper form and it's functional. So we moved this into some immunofluorescent models. On the left, you'll see some panels for immunofluorescence in vitro. So these are just in well plates. The blue is the DAPI for nuclei of the cells. The bottom two panels are a normal human dermal fibroblast on the left, so our control. The right is an RDEB, unmodified wild-type cell. And as you can see on the right, we're not seeing any anti-COL7 staining, which would be in the red. On the left, you will see some modest amounts of collagen 7. This is what our cells, fibroblasts, normally produce collagen 7 and contribute that to the basement membrane zone. But what we were interested to see was in the panel, these are two replicates of the same cell line as this. So this is the transduced version of these two cells. We saw actually hyperexpression of collagen 7, so quite a bit larger amount of collagen 7 being produced by our cells. So the now previously non-functional cells are now hyperproducing functional collagen 7 at a rate, and we also confirmed this with ELISA, then normal human dermal fibroblast line here. So again, since that's promising, we moved this into an animal model we developed with Stanford University, was shared with the FDA as part of our pharmacology preclinical program. And here are some results from that, an example. So what you're looking at here are cross-sections of skin. So basically what we did was we took RDEB patient cells, so keratinocytes and fibroblasts, which make up the epidermal and dermal layers of skin. We co-culture them into these, what we call skin substitutes, so they mimic human RDEB skin into a sort of a well plate look substitute. And then that was grafted onto the back of skid mice, and then we injected directly into those mice, into that graft, our product or the control. And these were the punch biopsies of those grafts, and then they were examined using histology with an anti-Col7 marker. And here's the results we saw. So as you can see in the RDEB lane frame there's no light up of collagen 7. The green you see in normal human dermal fibroblasts, this is what all normal basement membrane zones should look like, populated with anchoring fibrils. It's collagen 7 rich. What we were interested to see in our positive, though, in our product replicate was, all this collagen 7 expression is now being seen in the dermal layer. And what was really interesting that if you focus on this white arrow, we're seeing the early sort of expansion of the new basement membrane zone for this RDEB graft. So you can see it now, the IF of it reforming under the epidermal layer, above the dermal layer. So this was the pharmacology data that gave us confidence moving into the IND, which we filed with FDA and was actually allowed in April. So I can give you now an update on where we are on the clinic. After I talk a little bit about collagen 7 longevity, the idea here is we want this to be as long-lasting as possible, right? We don't want to be retreating patients all the time. So we have some anecdotal data for that. Our clinical trial is designed to look at this long-term. So obviously the cells are autologous. We already discussed about rejection risk. We directly administer this. There's been some attempts to do this intravenously with recombinant collagen 7. Unfortunately, saw problems with homing and toxicity doing it that way. The integrative nature of the vector allows us to, obviously express collagen if the cells do continue to divide. And the model I described on the previous slide was published by Stanford doing some work in keratinocytes, and they actually saw collagen 7 expression out to 12 months. And that was their last time point. They didn't see a drop-off in expression. So we believe that, based on these data, we have some hope of some long-term expression. Our clinical trial, which is described here and is on clinicaltrials.gov, also is designed to determine that. Not only we're evaluating safety, but we're looking at these mechanism of actions, which includes immunofluorescence and electron microscope analysis for morphology of the anchoring fibrils from 12 weeks, 25 weeks, and 52 weeks from administration. And then we're also doing 3D imaging of the wounds as well to look at wound progression, wound healing, wound persistence, and longevity of healing from 4 weeks out to 52 weeks. So we think this should give us a good look at the persistence data in the human subjects. This is a phase 1/2 study. We're looking to look at 6 adults. It's very rare to find adults in this population. Luckily, Stanford has a good registry for this. And then 6 additional pediatric subjects in phase 2. And we're currently have 4 patients enrolled. We started rolling in July. Lucky to have 4 enrolled fairly quickly. We're working on our last 2. And the manufacturing for this, as I mentioned at PCT, is ongoing. So we'll be looking to administer to our first patient sometime around the end of the year, at the end of this year, and then start following patients up after that through 2017. So that's our milestones that I just mentioned. We have year-end for first adult subject treated. First clinical data, third quarter of 17. We're looking to start reporting out these EMIF results and some of the wounding results, and obviously safety as well. And then in our 013 program, we're looking to be IND ready by the end of 17 as well. So I want to thank you all for listening and thank ARM for inviting us again, and the team at Fibrocell and our partners at Exton. And I'll be around if you have any questions. Thank you very much.